All right, so this I can statement video, learning target tracker video, whatever you want to call it, is all about RNA processing. So this is something that I didn't know about until I guess AP Bio or maybe even college. Um, I thought, DNA made RNA, RNA went to the ribosome, do, you got your protein and you're done. And that is how it happens in bacteria, but that is not how it happens in you. So when RNA is made in and this happens to not just mRNA. I'm going to talk about mRNA, but it also happens to tRNA, rRNA, those others. They have ways of being processed as well. But when what happens is when that mRNA pops off of the DNA, when it's been transcribed, the RNA polymerase reaches that termination sequence or the terminator sequence. Everybody falls apart. RNA polymerase goes and takes a nap. The mRNA is not safe. It's not in a structure or a form that is safe to leave the nucleus. If it left the nucleus right then, enzymes would just attack it and tear it up. And so what it does instead is it goes through this process called RNA processing. And there are 3 steps to that. The first step is that we add a modified guanine to the 5' end. We call this the 5' G cap, or sometimes just the 5' cap, or sometimes just the G cap or the guanine cap. But basically, it's a GTP, so think about like ATP, but with a guanine instead of an adenine. And we add that to the 5' end of the RNA. Then on the other end, the 3' end, we add a poly A tail. We add a bunch of adenines down there. Just, the red part of this is, A's, U's, G's, and C's. So it's a chunk of RNA. And so we're just going to add a bunch of A's to the end. That's going to help it get out of the nucleus, okay? It's going to help it avoid enzyme degradation. It's going to, in other words, keep enzymes from chewing it to pieces when it leaves the nucleus. And it's going to help it attach to the ribosome. So it's going to help it get out, travel safely, and attach once it gets there. The third and final thing that has to happen is RNA splicing. And so what we do is there are parts of the RNA that aren't needed, and we call these introns. What we do is we cut those out, and then we throw that away, and we fuse those exons. We don't throw it away. We fuse those exons together. What we do instead, we recycle it. So we break AT or AU, GC, AG, whatever, break that down into the bases and reuse it to make more RNA. And we fuse these exons together. So now what we've done is we've removed these pieces we don't need, and we have our final piece of RNA with the cap, with the tail, and all the introns removed. So I can explain the process of mRNA processing. I can explain the 3 steps of mRNA processing. I can explain the process of splicing. So that's just cutting 2 ends of something, removing it, and then fusing those ends together. And then I can explain alternative RNA splicing. So, and here's that spliceosome that you've learned about, the small nuclear RNAs joined with some proteins to make small nuclear RNPs or SNRPs. And that is what is going to cut that intron out, toss it to the side to be recycled, and then fuse those exons together. So that's like the little worker bee there. But a lot of people are like, why? That seems very wasteful. If we're just going to cut those introns out and recycle them, why don't we just not copy them? And the answer is because sometimes an intron in one case might be an exon in another. And so what we can do is we can splice it different ways. So in this picture, we have an exon 1, an intron, exon 2, intron, exon 3. What could be in a different scenario, maybe we need a different protein in which exon 2 needs to be cut out as well. We just need a protein that's made of exon 1 and exon 3. And so we would cut out exon 2, it would become an intron. So we would cut that out with the other introns, and we would just have the green and the purple. They would fuse together, and we would send that to the ribosome to make a shorter protein. And so you can see what it allows us to do is to have a lot more flexibility so that we can produce a lot more proteins from one section of mRNA rather than just one. And so the reason we have introns is because sometimes they're not always introns. Sometimes they're going to be left in as an exon, and sometimes exons are going to be taken out as an intron. And that just, like I said, it gives us a lot more flexibility, allows us to make more than one protein from one section of RNA, which is basically the same as one section of DNA. So I hope that helps, but if you still have questions, let me know.